Okay. So basically, this is a review of what you guys did on the pattern matching. So this is a generalized amino acid. Grab my pen here. So the R group, this is a variable, okay, and it changes. And like it says at the top here, there's 22 different protein-making amino acids. However, for organisms that use DNA, which is pretty much every living organism that we know about, and also viruses, which aren't considered alive, we only use 20. When we say they're coded for in the genetic code, like that goes back to when you had first year biology, where you had that table and you had to look up if you had A and U and G on your mRNA, then you had to figure out which amino acid it was, and it was methionine. That's what they're talking about. So the R group is unique. It can be as simple as just an H, as in glycine, or it can be really complex with like a ring or extra chains and stuff on it. You are not required to memorize the 22 different amino acids. Um, those, that would be like a upper level, graduate level, like college type thing, where they would make you memorize what they all are. What you do need to know, and this was on last year's AP exam, you need to be able to label the functional group. So you need to know that this particular group here is an amine group. It's the NH2 group. Sometimes it's written the other way around as H2N. It's the same thing. Okay, and then you need to know that this is the carboxylic acid group. So it's written as the COOH. We don't write CO2H because the COOH is telling us it's bonded in two different places on that carbon. It could also be written backwards as well, depending on, which way you're viewing the molecule from. It could be HOOC. Okay. Here's an example. This is methionine. That's the one I keep using as an example, and it's the one I remember because it's the very first one that's at the beginning of every single protein, like almost 100% of the time in living organisms. There are a few rarities that sometimes don't use it, but the majority, we use this methionine, and it's because methionine in its R group here, all of this is the R group. Okay, the R group has the sulfur, and the sulfur forms the disulfide bridge. Here's the backbone at the bottom. That's super important. So you have to have methionine. We also will come back to it when we talk about central dogma and protein synthesis in a later chapter. But the start codon on mRNA is AUG, and then that codes for methionine. That comes first. And it's kind of a large R group. Most are smaller than that. Okay, the smallest one is glycine, just has an H. Okay, I'm going to skip past glucose here because we already did those. Okay, this is an example multiple choice question on your exam. About half of it will be multiple choice, and the other half is going to be a short or free response, assuming we have a normal testing year. I don't know what May is going to look like. Last year, they ditched a multiple choice completely and did a modified short response. So if you were asked here to pick, hopefully you would remember what we just saw, which is the central, whoops, erase that. The central carbon was what we just saw. Central carbon with the carboxyl group. The amine group. All right, I'll use that line. I don't know why I did that. The R group at the top, and then the H coming off that central carbon. So if you remembered that, you could draw it out and you could say, oh, okay, then it is this one, D. This is actually a picture of glycine. But if you did not remember that, you could kind of look through it a little bit more closely and hopefully rule out B, because you see there's a sulfur here and the sulfur is not in the R group at the top where it's supposed to be. So hopefully you could rule that one out. A is really close. It's just missing an O here. So that one's kind of the distractor. And then hopefully you would remember that there's only supposed to be hydrogens coming off the central carbon in an R group. So hopefully if you didn't quite remember, you could for sure rule out B, maybe possibly rule out C. So then that way you would know. Okay, I'm gonna skip over that. These are back to carbohydrates again. Okay, so here, I like these little animations. It kind of shows you the condensation makes bonds. It takes small monomers, joins them, and makes polymers. And then hydrolysis is the opposite of those. So if you look here, we're talking about the proteins. Okay, the OH on the carboxyl and an H on the amine break off, and then you get a peptide bond. The peptide bond is a covalent bond, but it's a specialized kind. In organic chemistry, covalent bonds get special terminology. You do need to know that it's a peptide bond. Well, it's here. Okay. And you can see you also get the water. And this goes on and on and on and on. So you can get a polypeptide chain. Polypeptide chain could be hundreds or thousands, potentially, of amino acids long. This condensation hydrolysis does also happen with carbohydrates. You get a glycosidic bond. That's just extra information. Peptide, you have to know, okay? And then in fatty acids and glycerols, you get the same sort of reaction. So these OHs break off and the hydroxyl group here breaks off. You get a water, and this one's called an ester bond, but that is also extra information. It's the peptide bond that you need to know. Okay, and here's another diagram. It's better than the one I drew earlier. Okay, water comes out, water goes in. Okay, and these go in both directions. You do not need to know the enzyme names right now. Okay. Here again is showing the proteins and the water coming off a little bit more specifically. The water gets removed from here. Okay. Now, proteins were we actually started, humans started trying to crack the code of proteins well before DNA. In the 30s, I think we started on it, late 30s, and maybe early 40s, because World War II kind of started in the middle of there. And we worked on it, and we worked on it, and we worked on it, and we worked on it. And it wasn't until like the late 60s, early 70s, where we finally started understanding the structure of proteins from a biochemical standpoint. And the reason is, it turns out with 20 different amino acids, you have a huge number of different combinations that they can be. And the R groups are not regular, and it turns out the R groups interact with one another in very complex ways. So it's really difficult, versus DNA was started later, and I think the code or the shape was cracked around 1954, maybe 1956, somewhere in there. So they started on it later and they figured it out much, much sooner, but it's because there's only 4 different nucleotides, and we'll get to that next week. But the 4 different nucleotides means that it's much more regular in appearance, so it was an easier problem to solve. So polypeptides is another word that's similar to protein. It's not exactly the same. Um, a, a protein okay, why are you doing that? All right, back to the pen. So a protein has been folded. Let me try it down here. Versus a polypeptide is still a straight chain. Okay. A protein can have one polypeptide chain that is folded, or it could have more than one polypeptide chain that is folded. So for example, hemoglobin has 4 polypeptide chains that make it up. It has 2 alpha and 2 beta subunits. Helicase, the enzyme that unzips the DNA, that has 6 polypeptide chains in it. Okay, so you have infinite orders of possibilities with the 20 amino acids, and then you could also recombine the polypeptide chains in many different ways. So proteins are super complex. Okay, so these are just an example of different orders that you could have them in. And they're very, very much analogous to an alphabet. We kind of don't give the alphabet much thought because we use it all the time, but it is a fabulous invention, and really it has only been invented once. It's been modified and it's changed over time, but it was only invented once by the Phoenicians. Prior to the alphabet, we had more like pictographic languages like hieroglyphs or like Chinese or Japanese characters. Actually, Japanese characters, just they stole the Chinese ones, where those, each picture was a different word or concept or symbol. So it's very, very difficult to use for communication. It requires a lot of memorization versus our alphabet with our 26 letters in English. We can use those 26 letters in many different combinations and to make words, and then we can rearrange the words to make sentences, and those sentences can make paragraphs, and then those can make like books or movies or whatever, all out of 26 letters. The same goes for amino acids. With only 20 amino acids, you can have an infinite variety of proteins. Proteins make up about 50% of the dry weight of living organisms. So if you evaporated out the water, about half of what is left is going to be proteins. The other half is going to be a mix of lipids, carbohydrates, and nucleic acids. So they are really important for both structure, and also function. Okay, they do many things. So for structure, they make up the framework of a lot of the connective tissues. There's a ton of proteins, for example, running through a bone. It's not just the calcium salts. And also for function, they make up our enzymes that do all these chemical reactions, and they act as like transporter proteins on our cell membranes as well, or they can act as biosignaling molecules, biochemical signaling molecules as well. Okay, so if you were to break this peptide bond down, you have to add a water and split it, and then you have to restore this OH to the carboxyl group and restore this H to the amine group. If you see a problem like this, don't let this little thing freak you out. That's red. Oh, that was terrible. Let me try again. There, that's red R prime. Okay, that's just denoting that here's an R group and here's a second R group. We're not identifying which one it is. That's all that is. Okay, I'm going to skip over the monosaccharides. Okay. Monomer, dimer, and polymer. The mer is for molecule. It's generic. Mono means one, di means two, and poly means many, which in practice is 3 or more. Okay. You'll notice lipids aren't here. Okay. You do need to know for each of the 4 groups that we're going over, oh, and also DNA isn't here. For each of the 4 groups, you need to know what the monomers are, and you do need to know the polymers. So for carbohydrates, it's monosaccharide, disaccharide, polysaccharide. The difference is we change to saccharide, which is means sugars. But for proteins, the word is peptide. So we don't say monopeptide, that would be too easy. We say amino acid. But a dipeptide is 2 amino acids put together, and then a polypeptide is 3 or more. Dipeptides are useful and are used by the body as peptide hormones. For the triglycerides, this is your lipids. Lipids don't really have monomers in the same way, and they don't form polymers. But they do, the triglyceride is probably the most complex one, so you'll have an OH and an H come off and make a water, and it can do it 3 times. That part's just review. So then we say the triglyceride looks like an E. We just went over that already. All right, so I'm going to pause the record. Okay, so these are the notes, the section on proteins when it comes to protein folding. Here's some animations. I'll link these notes on the chapter materials. I didn't put these ones up yet, but I did put the previous ones up. So you could play around with those if you'd like. But basically, proteins, like I said, they're long chains of polypeptides, and they are folded in a very particular shape. If they're not in that particular shape, it's not going to work. Okay, that didn't work. The protein, whatever job it's doing, is not going to work. So there are 4 levels of protein structure, and thankfully they're named appropriately and they go in order. So it starts with 1 and it goes to 4, 1, 2, 3, 4. But we don't say level 1, 2, 3, 4. We say primary, secondary, tertiary, and quaternary. So tertiary is 3 and quaternary is 4. You may not have heard those terms before. And when we write it out, we write it like this, with a 1 and a degree sign. And you read that primary. So it's not a 0, it's a degree, kind of like if you have degrees Celsius. So when a ribosome makes a protein, it starts off with the primary structure, and it's just the sequence of the amino acids. So the ribosome and the mRNA and the tRNA all get together, and they put the amino acids in a particular order based on what the DNA told it. That is the primary sequence, and you can think about this as if these were letters in the alphabet. Like it's a very good analogy. Okay, so these are like letters. So this is the spelling of the protein. So if we have a protein that, so methionine, glycine, cysteine is a different protein than methionine, glycine, cysteine. Hopefully I said those differently. Okay, totally different. There's some exceptions, but mostly different. Kind of like how with words gray and gray, AY or EY can mean the same thing. There's a little bit of room for error with proteins, but we'll come back to that later. The secondary structure now is when you have hydrogen bonds form, and it's only hydrogen bonds that form amongst the backbone. Okay, so all that you had to highlight before, so the C and the COOH and I'm not going to draw in the others, okay? There was an R group there that's invisible. But this has electronegativity and this has electronegativity, so this hydrogen will actually form hydrogen bonds with other amino acids that are in that polypeptide chain. And there are, for our purposes, there are 2 main regular repeating secondary structures, and you do need to know them. So one is the alpha helix. So this is a Greek letter alpha. So it looks like a fish, but we pronounce it alpha. The other is a beta pleated sheet. So it looks like a bee with a tail. It's pronounced beta. And those are where we get the word alphabet from, alpha and beta. So the alpha helix, a helix is a spiral, screw-shaped. So the alpha helix, the way the hydrogen bonds form, it pulls, instead of being a long chain, it pulls the polypeptide into a spiral shape. Or the beta pleated sheet, it folds up the protein into kind of like a fan-like shape. So you need to know it's hydrogen bonds that are holding this into place. And also that there's two different shapes those hydrogen bonds can make. And within a single protein, polypeptide chain, there will be sections that are alpha helices, that's plural, and there will also be sections that are beta pleated sheets. And they follow rules in how they form, but one single polypeptide chain can have multiple alpha helices and multiple beta pleated sheets. You're not going to be asked to fold a protein, okay? It's super complicated. We don't yet know all of how every protein folds. It's part of why we haven't fully annotated the genome yet, is because we have to figure out exactly how these proteins fold up. The next step, the secondary step, this would be if primary is akin to letters, then secondary is akin to words. Okay, so not just the spelling, but where are we putting the spacing? If you had a whole string of text with no spaces and no punctuation in it, if you didn't have a period to know when the sentence ended, it would be very difficult to understand. So this is like words with space, and also punctuation. Now tertiary, when we get to tertiary, this is when you get to like the sentence level. So you have a grammatically correct unit that has a subject and a predicate, maybe even a direct object or an indirect object. Okay, but it has a meaning to it. Apologize for the handwriting. Okay. So the tertiary level, this is still only on one polypeptide chains. Okay. This is where the R groups come into play. So the tertiary level is what's giving our computer scientists and our proteomics people fits, is because the R group is so complex. So in general, what will happen is the middle of a protein is going to be the R groups that are hydrophobic. And the outside is going to be all the other R groups that are hydrophilic. And the reason is, in living organisms, we're just bags of water. So within the cytoplasm and the cytosol, it's mostly water. And outside of the cells, like in your bloodstream, for example, blood is mostly water. All the fluids within bodies that's not in a cell is still going to be water. So the water forces the hydrophobic parts to interact with each other and form a core, okay? And those hydrogen bonds are also going to help keep that in place. Now, they didn't list this out, but the tertiary, I'll try to write it here. It's held in place by hydrogen bonds. It's also held in place by those disulfide bridges we talked about. I'm not listing these in order of strength of bond. It is also held in place by ionic bonds. Some of the R groups do ionize in water, and then the positive ones and the negative ones will attract. And then also hydrophobic interactions. If you're in upper level chemistry, that's also called van der Waals forces. Okay, so there's a lot of different bonding types because there is such a variety of amino acids. Okay. When you get to the quaternary structure, the bonds or the bonding type that holds it in place are the same as when you do the tertiary structure. Okay. Quaternary structure. This is when you have two or more subunits, or another word for that is polypeptide. When it's a single, it's a polypeptide, but once you combine them together, then we typically call them polypeptide subunits, but we oftentimes drop the polypeptide part. So this would be, in the same analogy, this would be like paragraphs. So you don't just have single sentences, you put the sentences together, make a paragraph, and you can put some more paragraphs together and make a whole essay if you want. There are some of both. Some proteins stop at tertiary structure. A lot go on to quaternary structure. There's even a few that they stop at primary or secondary, and then an enzyme kind of cuts them apart, which seems kind of weird. Like that's how insulin is made. Like why would you make a long polypeptide and then cut it into little bits? But that happens. And then the primary or secondary ones that get cut into bits, those ones will be hormones. But the true proteins, those are going to have tertiary and oftentimes quaternary structure. All right. Okay. So here it is spelled out a little bit more so that you can see it a little bit more slowly. I do like these animations that are embedded here, so I'll link it so you can practice with it. But with the polypeptide primary structure, proteins do have varying numbers of amino acids, just like words have varying numbers of letters in them. Typically, like an average protein might be in the like 100 to 300 or 400 amino acids long range. But we do have one, our largest known protein, Titan, I believe, is in the thousands. So it just kind of depends on what that protein's function is. Okay. This is just a review here. We've already talked about that. Here are all 20 amino acids if you were curious. Whoops. You tend to see them organized into a chart like this. Each one has its own unique properties. So if you're an upperclassman in college or a graduate student in college, if you're going into sciences, you would have to memorize the chemical structures for all of these amino acids and what their properties are. You don't have to do that now, but you should know the highlighted parts here. So some are going to be polar R groups, some are nonpolar, some will ionize, so they'll be a full positive or a full negative charge, not just a partial one. And then some contain sulfur. Okay, and those are the four properties that determine how that protein folds. Okay, here's a close-up again on the alpha helix and the beta pleated sheet. This, you may see pictures like this in your text, and as we kind of go through, if you look on the protein database, you can just Google that. You can see like every protein that we know its structure and it'll be represented that way. Or you can change it and do a space filling model. This is called a ribbon model. We use this rather than the ball and stick model when we're doing proteins because their proteins are just freaking huge. There's so many atoms if you were to do them out atom by atom. It's just very tedious, time consuming, and a lot of those atoms are repeated with the backbone. So we tend to draw it like this, where we'll show the beta pleated sheet, we'll show the alpha helices, and then see how they fold around each other. These green areas, we don't have to know their structure now, but it's a hairpin turn, basically. This is a small one. Okay. Here's more complicated ribbon models. You can see here and you can see here. So you might see these different spirals, and we tend to color code them too, based on what each one's function is. Here's a second one, and you can even kind of see in this one, this is probably an active site here. Okay, you have a little space there. And then if this were an enzyme, this may be an allosteric site over here, or maybe here. The hydrophobic part is going to tend to stay in the middle unless it is a plasma membrane protein. If it is a plasma membrane protein, then the parts that are embedded in the plasma membrane that's hydrophobic will be hydrophobic. So it may indeed be on the quote outside of the protein, but if it's up next to other hydrophilic parts, it's okay. Like it'll be stable chemically. Okay. This is just an example, a very, very small example to show you. So if these are our amino acids here, here's the names, here's their abbreviations, and then here's their properties just briefly, positive, negative, sulfur, nonpolar, polar, then here's a primary structure. And if you were to read this, you'd say serine, 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 lysine, valine, valine, okay, and on. And notice you can have repeating subunits, that's okay. But then if this were going to fold up, it will follow those rules in order: polar outside, nonpolar inside, unless it's a membrane protein. And then positive and negatives go together, and then disulfide bridges. So a folded polypeptide chain would look something like this, where you have the disulfide bridge, you have an ionic bond there, the hydrophobic parts in the middle away from the water, and the hydrophilics on the outside. And again, this is super, super simplified. For one, it's 2D. Water's all around it, so these will actually fold up in a spherical shape. And then here's quaternary structure, so you can see. Okay, so here's an example of a hemoglobin. This is the ribbon model. This is kind of more of a cartoon stylized version, but there's 4 polypeptides in it, 4 subunits. And you can sort of see them here. There's 2 different shades of blue there, and then a green and a teal here. So this is hemoglobin, and it actually has 4 functional, or well, 4 oxygen binding sites. 1, 2, 3, 4, 1 for each subunit. And then you have just a whole ton of these packed into your red blood cells to carry the oxygen. So you have 4. Okay. For people that have sickle cell anemia, they have a mutated version of hemoglobin that affects 2 of these subunits. 2 are alpha and 2 are beta. We will come back to that. That's one of our they used to be called illustrative examples, but they've changed the term for it now. But anyways, it'll still be on the test. Hemoglobin is the reference protein we always go to because it was the very first one we figured out the structure of. And it took us a long time to do it. And they were looking into the hemoglobin because there do exist various mutated forms of hemoglobin, not just the sickle cell one, but others, and they all cause anemia, and they're huge health problems. So that's why we were looking into that one. This one's an example of a DNA polymerase. This is an enzyme that makes new DNA strands from a parent strand. So you can see an active site here quite well, okay? And then this one is showing you an ion channel and a plasma membrane. So you would have subunits on this side and on this side. And then the parts in the middle where the ions would go through, those are going to be hydrophilic. But then the parts that are embedded in the plasma membrane, those are going to be hydrophobic parts. You do need to know functions of proteins, like how they work in living organisms. So we tend to classify them in 2 different ways based on how their structures look. Fibrous looks like a fiber or a line. Globular looks like a glob. The globular proteins tend to be the ones that are functional. They do well, they all do a job, but the functional ones tend to be more like mobile, working, going throughout the cell in the body. Fibrous ones tend to be more structural. They stay in one place. They do have a function, it's for support. Okay. But these ones are the enzymes and these ones are support. So fibrous, a good example is keratin. Keratin fibers can be arranged in many different ways. That's what makes up your fingernails, also your hair. Whether you have straight hair, curly hair, kinky hair, it's all keratin. It's just arranged in a different way to give it those different shapes. It's also the same thing that's found in hooves on cows or horses, or the horns on rhinoceros. Some horns are bone, but the rhinoceros one is keratin. So it can go from like not very strong, like if you have brittle hair, to super strong, like a rhinoceros horn. There are elastin fibers that can be found in the skin, for example. Those are stretchy. And then there are also collagen fibers, and that's for strength. So collagen, for example, we were talking about bones earlier. Collagen is found all throughout the bones to give a scaffold for the calcium salts to kind of glom onto. So that bones are strong, but they're also flexible in a way too. The globular proteins, these are more functional typically. So there are tons of enzymes, like there's digestive enzymes. Amylase is an example. We just talked about DNA polymerase as an example. Helicase is an example. Anything that ends in ase. They could also be hormones, like insulin is a hormone, regulates your blood sugar along with glucagon, another hormone. They can be transport molecules like hemoglobin. Hemoglobin is super important. All animals have a version of hemoglobin, whether you are a planaria or you are a spider or a people. We all have some version of hemoglobin, and we've actually found that even some plants have hemoglobin, so it has a very long ancestral history. And there's like, oh gosh, I think at least 6 different versions of it. Like we have 6 different genes for it as humans. Fetal hemoglobin is different from adult hemoglobin. It's a whole big thing, and it has a really fascinating evolutionary history. Proteins can also act as antibodies. Think about like antibiotics, similar thing. So antibody is the job that it does. A specific protein is an immunoglobulin, is an example of an antibody. And what these do, if a person is sick with a pathogen of some sort, is an antibody sticks to that pathogen. Like it'll glom onto, say, the outside of a bacterial cell, flag it for the immune system to know, like, hey, that's the bad guy, come kill it. And it'll also slow it down too, so that the microorganism can't go very far. Excuse me. And then lastly, they can also be used for transport across a cell membrane. So the sodium-potassium pump is probably the most important one, at least to our curriculum. The sodium-potassium pump is what regulates our nervous system and also our muscular system. And without those, we would die, basically. This is just some more modeling to kind of show you. So here's the ribbon model again. This would be a space filling model for an enzyme where you can see the atoms kind of like stuck together like balloons. Okay. I've already said that. Okay, and that is the end of the notes.